Long before we were ever on this planet, bacteria and bacteriophages were waging a battle, an arms race against each other as to who would survive and who would die. And so the bacteriophage was always trying to inject its DNA into the bacteria, and so the bacteria, to prevent this, developed ways to chop up that viral bacteriophage DNA or phage DNA, and it would survive, whereas the alternative was the phage invades, replicates itself, and the bacteria dies. The bacteria was able to restrict the growth of the phage with something called restriction enzymes. We now know them as restriction enzymes. What they did was they chopped the DNA of the phage into tiny pieces, but it wouldn't chop up its own chromosomes, and it wouldn't necessarily chop up the DNA of every phage, just the phages that it had kind of evolved with. There are so many different types of restriction enzymes that we've discovered now in different bacteria, and each one cuts at a different site on the DNA. These sites are normally palindromes. They're like 4 to 6 nucleotides long, and they're going to read the same way on either strand. That's what a palindrome is. So looking 5' to 3' on each of these strands, you're going to read AAGCTT. Some of the enzymes that would recognize these sites would cut straight through the double helix, creating what we call blunt ends, but some of them that we discovered later on would actually cut a jagged line, leaving what we call sticky ends, meaning that you could cut 2 different pieces of DNA at the same site and recombine the pieces together. So you could start with something like this and end up with something like that. And we call this recombinant DNA. Often the DNA that we're using to do this is from 2 different organisms. And we've gone way beyond this now. We don't even need the restriction enzymes that make sticky ends. We have enzymes that can add on little like A tails and T tails so that they'll stick together. But this was where it started, and this was what we did for quite a while. Often, and still to this day, we do this with bacteria because they have little circular plasmids. So they've got their large circular chromosome, and they've got lots of these little ring chromosomes called plasmids that can be cut with the same restriction enzymes as whatever DNA you want to target. And so you cut that with the restriction enzyme, and then you can insert your target DNA into the plasmid. This turns that plasmid into what we consider a vector because we're going to use this to transform the bacteria. We're almost like we're infecting the bacteria, but we're moving our target DNA within this plasmid into a host cell, and that cell, the vector in that cell, will replicate itself, and the cell will replicate, and so we'll have many, many copies of our target DNA. Anytime you put a new genetic piece of information into an organism, we're going to call it transformation. And so this is bacterial transformation with this vector. There's a lot more I could tell you about vectors. They've got some special genes and regulation sequences, but we're going to stop here for this class. So what is putting these tiny target pieces of DNA into vectors and then into bacteria, what has that really done for us as humans? Well, it's done a number of things. It allows us to produce things that humans need, like insulin and clotting factors, vaccines. So any protein-based product that could be the end result of a gene that we put in that target DNA sequence, the bacteria could produce for us. We can create crops with all sorts of desired traits, things like sweetness or fungal resistance or herbicide resistance. And if you've heard of those infamous genetically modified organisms or GMOs, this is exactly what we're talking about. We can put a vector into a bacteria that is known to infect a plant, and you can actually move that gene and that protein product into that plant and have the plant express it. So again, we're talking cross species here now, and not always plant to plant even, sometimes bacterial to plant. So we're doing some crazy things, and it's done a lot of good. It feeds a lot of people, but there's still a lot of debate about it. And then we've also got a growing number of gene therapies that we're doing with this technology. So there's so much deeper you can go into this if this is something you're interested in. Of course, to do all these amazing things, you need DNA from a particular gene. So it actually makes sense to work backwards. You can find the protein you want, and you know that protein is made from mRNA, and that mRNA is found in the cytoplasm. There's even ways to go in and probe for that mRNA, because all messenger RNAs have those poly A tails, so you know what your target is, what you're looking for. And really, mRNA is a lot more abundant and readily accessible than the DNA is, and it tells you a lot about what the cell is doing, because that's what the cell is expressing at any given moment in its life cycle. And by about the 1970s, we also knew that there were viruses that were using RNA, not DNA, as their genetic material. But there was still DNA taking over the host cells. So the viruses had RNA in their mature form, but somehow they were taking that RNA and they were turning it back into DNA. And so the very first thing that this was called in the first primary papers that were published was that the central dogma was being reversed, that RNA was being turned into DNA instead of the other way around. We call this reverse transcription now because we flipped that transcription step, the DNA to RNA, rather than the RNA to DNA, or RNA to DNA rather than the DNA to RNA. The enzyme responsible for this process we've aptly named reverse transcriptase, which we discovered a little bit later. And you can go into a lot of details with this process, but here's the basics. You throw your mRNA into a test tube, so you've already isolated it. I skipped that step. You attach a primer of T's to match up with the poly A tail. You would add the reverse transcriptase and allow it to add to that 3' end. Then you add an enzyme that's going to consume the RNA and allow DNA polymerase to start filling in those blank spaces, and then allow that to continue, and you have your complementary DNA or cDNA sequence. We call this cloning sometimes. There's a lot of things we refer to as cloning in the biotech world. This is just one of them, because we're making a copy of some piece of an organism's DNA. We have lots and lots of cDNA now. We've made libraries out of it. We know what DNA or what RNA was being expressed in different stages of a cell's life cycle. And so this was an early step in, taking these biotechnology tools and letting it be like a learning tool as well. So up to this point, we've got two tools in our molecular biology toolbox. We've got a process for making complementary DNA from RNA, and a process for taking segments of DNA and creating recombinant DNA from one organism, or more often many organisms, that we can have expressed by another organism like a bacteria. So we can make lots of human insulin without having to take that insulin from a human. We could take it from another organism.